Ratna and Dr. Ferry, can we start?
Yes, sure.
All right. Okay. Good afternoon, everyone. My name is Dr. Wu from Hospital Seberang Jaya. I'll be your moderator for today's presentation. On behalf of MMIG, I would like to welcome all of you to our webinar.
Dr. Janyan, I think have to unmute yourself. Dr. Wu.
Sorry. Can you hear me? Can you hear me?
Yes. Can.
Okay. I think I better start again. Okay. Good afternoon, everyone. I'm Dr. Wu from Hospital Seberang Jaya. Okay. On behalf of MMIG, I would like to welcome all of you to our webinar today. Okay. Before I continue, kindly mute all your microphone, and participants can post all your questions in the chat box, and we will answer the questions after each of the presentation. So we are very fortunate today to have two experienced speakers with us. Okay. First, may I briefly introduce to you our very first speaker. She is Puan Ratna, a Senior Scientific Officer in the Mycology section in Infectious Diseases Research Center, IMR. She has vast experience in mycology, including being a lecturer and an avid researcher. She is responsible in starting the PCR test for fungal identification in IMR. Puan Ratna has published numerous journals in mycology, and she's also a reviewer for international journals. So please welcome Puan Ratna to give her presentation on characterization of common yeast. Can you all please mute your microphone? Okay, please welcome Puan Ratna to give her presentation on characterization of uncommon yeast pathogen identified in Institute for Medical Research, IMR. Puan Ratna.
Hello, hi everybody.
Hi, Puan Ratna.
Can you hear me very well?
Yes, very well.
Can you see my slide?
Yes. Please mute. Okay.
Okay. Can I start now?
Yes.
Okay. Today, I would like to share my experience on characterization of uncommon yeast pathogens identified in IMR since we established our PCR for fungal pathogen in 2013. So I won't share everything. I will choose a few fungals only, few yeast only. Okay, for introduction, as I mentioned before, molecular identification of fungal pathogens in IMR has been established since 2013. And 90% of the culture were common pathogens and 10% of them were uncommon. So therefore, in 2015, I proposed a project entitled Molecular Characterization, Antifungal Susceptibility Pattern and Taxonomic Study of Uncommon Fungal Pathogens for identification of this uncommon species. So the objective of the study is to study the uncommon fungal pathogen by phenotypic and also by
Yang ni kalau sekejap, yang ni kalau macam ni maksudnya memaafkan. Yang Yang ni mute ni suara dia, bukan suara kita. Oh, betul.
Puan Ratna, I can't hear you. Puan Ratna?
Okay, can you hear me? Can you hear me?
Yes, now can. Okay.
These are few species, uncommon species that I identify using molecular methods. Among them are Candida hemolyni complex. In this complex, there are few species, including a new species among this complex, which is Candida vultuna. And others are Candida hemolyni related species, for example, Candida pseudohemolyni and Candida auris. And we also identify few other Candida species which is uncommon, Candida versatilis, Candida wangnamkyomensis, Candida muscifera, Candida thier, Candida nivariensis, and Loderomyces elongatus. And also we identified some basidiomycete yeast, which is cause infections in immunocompromised patients. Among them are Pseudozima ephidis, Pseudozima antarctica, Pseudozima para-antarctica, Pseudozima sucubensis, Albomaniaca, Triodiomyces triodiae, and also very important species for me is Ferredonia cargensis. In this presentation, I would like to share few species which is I interested with. One is Ferredonia cargensis, secondly Candida auris, thirdly Candida vultuna, and last one is propose a new species is Wicker hamela species. Ferredonia cargensis, first isolated from the blood of HIV patient. This patient from Queen Elizabeth Hospital in Sabah. This patient was initially treated with amphotericin B, but clinical condition did not improve with the amphotericin B, and then it was changed to itraconazole, and the condition was improved after this itraconazole treatment. And then second case of Ferredonia was isolated from pleural fluid of kidney failure patient in Sultanah Fatimah Specialty Hospital in Muar. And this patient, the clinical condition of also improved with fluconazole treatment. And after that, I isolated and identified 11 more cases from different sources of clinical specimen, including blood, bone marrow, urine, skin, nail, and vagina swab. This is how Ferredonia looks like on the media, on the separate dextrose agar. At 48 hours culture, the presentation is dry and very wrinkled, and slightly hard, difficult to make a smear from this culture. And if we incubate this organism longer, at 72 hours, this organism start producing melanin-like pigmentation, and it become more darker after 5 days of incubation. So you can see at 120 hours, the colonies looks like darker. And this is how Ferredonia looks like under the microscope at 40 times magnification on yeast malt agar, and also on corn meal agar. They produce pseudohyphae, and these pseudohyphae are long and also branch. If you look at the 100 times of magnification, we can see the cells are elongated, irregular in size, and also they propagate bipolar. So when we try to identify this organism using other method, other system, using API20C, the organism is identified as Cryptococcus neoformans with high probability. With the VITEC2, also give different identification, Cryptococcus laurentii at 89%. And last time when I did the multi-top using the Bruker system, there's no identification for this organism. So we do the molecular identification using PCR sequencing method. Two set of primers that I use, ITS region for ITS region and for D1, D2 regions. And both give the similarity, very high similarity to the reference strain IPRC M30116. This is the type strain from Iran. They isolated the Ferridonia from the plant remnants. So when I make a never joining method, bootstrap test at 1000 replicates using Tamurane method, this organism is at a different clade with others organism which is related to. And here I list about more than 10 cases of Ferridonia from the different clinical samples. There are variables of specimen here, as you can see, and also there are few researchers already found this organism, but not from the clinical specimen, but they found it from the plant remnants and environment. So this is interesting because only in Malaysia we found it from the clinical samples, whereas others did not find it from the clinical samples. So we need to study more details on this organism and how this pathogenicity occurs. Sorry. Jumps to the end. Okay. The antifungal susceptibility testing of this organism show that this organism did not respond to the amphotericin B and fungins groups, but quite sensitive to azole, fluconazole, and itraconazole, but not so good with voriconazole, especially for the strain number 2. And we published these findings in Mycopathology. And the next organism that I would like to share with you is on Candida auris and Candida vultuna. I purposely compare this organism because these two organisms has a similarity. Can we remove it? As we know that Candida auris is a serious global health threat that been, CDC has already mentioned. This is a very emerging fungus that causes a serious global threat because of 3 main reasons. Because they are multi-drug resistant yeast and difficult to identify using the lab standard method. And then the misidentification can lead to inappropriate management of the patient. And this organism also cause outbreak in some healthcare settings. However, Candida vultuna, there's up to 2019, I've collected and identified about 16 cases of this organism that cause candidemia. And also this Candida vultuna difficult to identify using conventional method. Also using VITEC and multitop, they cannot differentiate this organism. this Wilsona also very related, closely related to Candida auris, and it has similar criteria with Candida auris, which is they are multi-drug resistant and difficult to identify, and we don't know whether this organism can cause outbreak or not. This is a comparison between the auris and Wilsona. On the Sabrot dextrose agar, they look similar, but using the chromogenic agar, this auris can produce light pink colonies. However, for Wilsona, they're not producing any pigment. Yeah, it's not moving. This is the microscopic presentation of auris and Wilsona. We cannot differentiate this organism unless we do some culture on the yeast extract agar and do the pseudohyphae production, which Candida Wilsona can produce pseudohyphae in YEA agar. Some system, for example, API 20C, Vitex, they cannot identify both organisms correctly. Which API 20C give Rhodotorula glutinase for Candida auris with high probability. And also Candida Wilsona was misidentified as Candida lusitaniae with also high probability. So Vitex also, I understand that some cannot identify this Candida auris and Wilsona correctly because they give Candida hamiltoni for both organisms. So the best method for me is to do molecular identification using ITS and D1, D2 regions. This is give the correct identification for both Candida auris and Candida Wilsona. However, Malditov for Bruker system, they cannot identify Candida Wilsona and give Candida Wilsona as Candida dobos hamiltoni. This is the antifungal susceptibility of Candida Wilsona and Candida auris. As we know that Candida auris is a multi-drug resistant, so the susceptibility show resistant to most of the common antifungals. Also for Wilsona, there are resistance noticed for fluconazole, itraconazole, voriconazole, and also amphotericin B. However, it's quite interesting that fungin group gives a sensitive effect to this Candida Wilsona. So this is the paper that I published with Prof. Matthias regarding the first isolation of Candida Wilsona. And also this is the first paper on Candida auris in mixed infection with Candida tropicalis. And last species that I would like to share with you is proposed as a new species, new strain, which in the genus Vica hamela. I isolated 3 strains of this species. One from the 67-year-old male with diabetes mellitus and hypertension, admitted to warded to the hospital because of hyperglycemia symptom, and he acquired pneumonia. And then patient 2, 25-year-old female with short bowel syndrome, admitted because of the high fever, high grade fever. And patient 3, was the neutropenic patient, mixed infection with Candida tropicalis. So this is how the Weaker Hamela looks like on the Sabrodextrose agar. They are similar like other Candida species, cream-colored, smooth, glabrous, and also yeast-like colonies. And when we culture on the SDA and YM, when we do some culturing on YM, they produce a pseudohyphae with branch and blastoconodia on the pseudohyphae. So this is the phylogenetic tree of Weaker Hamela with other species. As we can see here, this tree strain at the different clades with others. Thus, these strains are a new species of Weaker Hamela. The antifungal susceptibility testing, only caspofungin, they show resistance to caspofungin. With other antifungal, they are quite sensitive. So this is how we do analysis to confirm that this is a new species. They are not similar with any species of Weaker Hamela. Thus, we need to do some study to confirm this of new species. With that, I end off my presentation with the conclusion that multi-drug-resistant, novel and uncommon yeast species has been isolated from clinical samples, and they can be identified using PCR sequencing method and also using the advanced identification system. With that, I thank you all for your attention and selamat berbuka for those fasting today.
Thank you, Puan Ratna, for the very interesting talk. We will keep the questions to the end after the second speaker, okay? So you can post your questions in the chat box if you have any. Now we move on to our next speaker. Our next speaker is Dr. Ferry. Dr. Ferry is currently the group leader of the Department Medical Mycology at the Westerdijk Fungal Biodiversity Institute, Utrecht, the Netherlands. He has a distinguished career in mycology. He's an avid researcher. He was a board member of the EXMIG in 2021. And it's also a current member in various medical committees. He had received numerous awards in the field of mycology. So ladies and gentlemen, please welcome Dr. Ferry to give his presentation on molecular tools for rare yeast identification and outbreak investigation. Dr. Ferry.
Thank you very much, Dr. Wu, for your very nice introduction. So I'm Ferry Hagen from the Netherlands, and the title of my talk is Molecular Tools for Rare Yeast Identification and Outbreak Investigation. But of course, this also can be applied to the non-rare yeasts. So I've split my presentation into several parts, and I will start with molecular identification. And so now and then, I will talk about Candida auris and Candida fraterna will also be mentioned, as Dr. Ratna just discussed. So since the 1990s, there was the search for the molecular marker to identify fungi, and various molecular loci were used, and most of them were on the so-called ribosomal DNA locus, and that consists of various parts, so-called 18S, 26S, and the ITS, or it's the abbreviation for intergenic or internal transcribed spacer. And next to that, RPB1, 2, and TEF1 genes are used for the phylogeny of fungi. A few years ago, well, actually a decade ago already, a very big consortium investigated which molecular marker is the best to study fungal diversity. And they focused on 4 loci, the ITS, the large subunit, the small subunits. So that are the 3 ribosome markers and RPB1. And as can be seen from the right figure, the highest success was, PCR success was reached with the ITS locus. And since then, ITS has been the marker of choice. So here I've provided an overview of all the primers that have been developed to amplify specific parts of the ribosomal DNA. So the Luzzoni lab in the US has made this figure, and I adapted it a little bit. Several parts are highlighted. So we have the internal transcribed spacer that is nowadays commonly used for fungal identification, but also the large subunit is marked with a box, and the intergenic spacer. So the ITS and the large subunits are commonly used for Ascomycetes yeast, and the intergenic spacer is commonly used to identify Basidiomycetes yeast like Cryptococcus and Tragosporum. In 2015, a very nice study was published, and it's about the curated ITS reference database, and it was led by the ISHAM DNA barcoding team. And they also generated a lot of data for all the clinically relevant fungi, and they are accessible via the isham.org website. So you can blast easily your sequence there and then reliably identify your species of interest. And there is also a kind of algorithm provided with this paper. So when you blast your data against this website, against the database, you compare your ITS sequence versus that of the ISHAM ITS database. And if you have an identity that is more than 98.5%, then you have a reliable identification. If it's less than 98.5, then you need to do further testing or send the strain to a reference mycology lab. So many people use the database of NCBI, but it has a disadvantage that everybody can put his or her data in this database, and with that, it's not that reliably anymore. So I absolutely want to focus on the ISHAM barcoding database. And if there is a new species identified that is of clinical relevance, then it can be included in this database. And here I've provided an example of a sequence that we had from a believed Pigia Cactophila strain. But as you can see, it's below the 98.5% threshold. So probably we are dealing here with a new species, but we are investigating that. In 2010, there was a very nice study published by the group from the Mycotic Disease Branch of the CDC. And they focused on formalin-fixed paraffin-embedded tissues. And that's quite a difficult material to work with when it comes to molecular identification, not only for fungi, but also for bacteria and viruses. And this has to do due to the degradation of the DNA due to the use of the formalin. So in this study, Munoz Kadafic and their team, they use 5 commercial kits that are, well, at that time they were globally available. And what they did, they took more than 80 formalin-fixed paraffin-embedded tissues and checked if there was still fungal structures visible, and then split these samples in over the 5 DNA extraction kits, and they added also an additional lithic-case treatment to completely lyse fungal cells. So in total they had 81 clinical samples, and they scored them with the Gomori methemanine silver for if they are positive, yes or no. And what they did was they included a housekeeping gene, so that was a human DNA target, and they run 3 different kinds of ITS PCRs. So they are commonly used in the clinic, and that is the prime combination ITS1 with ITS4 that generates a fragment of 400 to 600 base pairs. ITS5 and ITS4 also 400 to 600 nucleotides, and ITS3 primer with ITS4 generates a fragment of approximately 300 base pairs. And what you can see in this graph in the middle is that the smaller ITS fragment of 300 nucleotides has a higher success rate when it comes to fungal identification from formalin-fixed paraffin embedded tissue. And at the right side, I provided a figure from a Dutch study. It's not specifically focusing on fungal DNA, but in this study, they described a so-called ladder PCR to check the quality of the extracted FFPE DNA. And as you can see from these gel pictures, you see a smear of DNA in the samples and also different success rates for the various samples. So some have fragments up to 400 base pairs visible, but some have even 0. So then I come to my favorite part, that's molecular typing, and I split this into fingerprint-based and sequence-based. So the first technique that I would like to highlight is amplified fragment length polymorphism, and I've split this slide into 2 pieces. Also because AVLP is performed in 2 steps. So the first step is digesting DNA of the yeast with 2 different enzymes, a frequent cutter, in this case MSC1, and a rare cutting enzyme, that's EcoR1, and that gives 3 different kinds of fragments. So approximately 90% is on both sides digested with MSC1, approximately 10% of the fragments is digested with EcoR1 and MSC1, and the remaining more or less 1% of the fragments is digested on both sides with EcoR1. And then you want to detect the unknown DNA fragments, and you can do that rather simply by adding a known sequence, the so-called adapter. And by adding the adapter to the unknown sequence, you also change the restriction site, so the DNA cannot be digested again. So this is the first step. So it's called the digestion ligation reaction, and it takes 1 hour. And thereafter, you have to run a simple PCR that includes a fluorescently labeled primer that binds to the rare cutting enzyme adapter. So in our case, EcoR1, and a non-labeled MSC1 primer. This because you want to detect on a capillary sequencer only the approximately 11% of fragments in your mixture, because if you add the fluorescent label to the MSC1, to the frequent cutting adapter site, then you end up with a smear and you will have no fingerprint at all. So this is a very nice technique to rapidly identify a large cohort of strains. And I show you some examples from the past 20 years. So at the left side is a dendrogram of Candida strains from a Dutch study in which we were involved. we were interested in which species of Candida were present in a sample set. So you can rapidly identify the strains by running an AVP and only selecting one strain per cluster and sequence it. And then you can automatically adapt the ID to the whole clade. This was how it was performed 2 decades ago. And nowadays we use AFP fingerprinting primary to see if we are dealing with an outbreak, yes or no. And also a very interesting approach was by using different molecular techniques to identify the species in the Candida hemiloni species complex prior the global outbreaks of Candida auris. So this was a paper from 2012, just before the first outbreaks were described. You can see that Candida auris can be easily differentiated from the other species. And in the right corner is a very large AVP tree with the various colors. So the clusters are colored orange, yellow, and blue. That identified at that time 3 different clades of Candida auris from very geographic regions, along with species from other species from the Candida hemiloni complex. Another advantage of using AFP fingerprinting is that you can easily identify hybrid species. In this case, I have some examples of hybrids between Cryptococcus diniiformans. So that's the old Cryptococcus neoformans variety, neoformans serotype D, that formed in a hybrid with Cryptococcus gutti, and also Cryptococcus neoformans serotype A and with Cryptococcus gutti. And interestingly, those strains were very weak positive with carnitine glycine bromothymol blue medium. So it took more than 2 weeks to become fully blue, while normally it's within 1 or 2 days the case. So we saw some, that hybrids have some decreased fitness compared to the parental strains. Similarly, AFP has shown to be useful to identify hybrids in the Malassezia furfur species complexes. And this has been very recently published in MBio by my colleague Bart Thelen and Teun Boekhout, where they also applied genome sequencing. The other technique that we frequently use is microsatellite typing. And it's actually a rather straightforward technique, but what you need is a genome sequence. And after you have that, you can run a very easy tool that is called the Tandem Repeat Finder. It's freely available on internet, and that identifies all the microsatellite loci in the provided genome. And we are specifically focusing on the DNA repeats that consist of 2, 3, or 4 nucleotides. So in this example, you see a sequence, an old school Sanger sequence of 28 times a repeat of CA, 90 times TAG, and 16 times TTCT. And the shorter the repeat unit, the higher the chance that different strains have a different number of repeats for that given loci. So you can multiplex this approach by adding a fluorescent label to each of the primers for each loci. So then you can lower the costs for PCR and fragment analysis. And in this case, I give an example how in the ideal world the microsatellite fragment peaks look like. So you see some stutter peaks and the highest peak, so the highest fluorescent intensity demarcates the real size of the fragments. And based on that, you can derive the number of repeats. And this is implemented in the software of the fragment analyzer. And so what you will end up with an overview of number of repeats per loci. This is indicated at the left side of this slide, and it's a kind of telephone number. And using a software tool, for example, Bionumerics, you can generate a visual representation of the genetic relatedness of all these strains. One such an example is that we applied microsatellite typing to a small cohort of cryptococcus patients from Slovenia. So we applied A4P, and we found, for example, one hybrid species. And we applied also microsatellite typing specifically for Cryptococcus neoformans sensu stricto, so serotype A, and Cryptococcus D neoformans, the serotype D strains. As you can see, microsatellite typing was able to identify all the loci in the hybrid strain. And this is quite a challenge if you want to do this by Sanger sequencing, because you need really specific primers for each of the MLST loci for Cryptococcus. And moreover, with this technique, we also observed that some patients that had cryptococcosis for a very long period, that the Cryptococcus strain evolved over time. So the microsatellite, the number of microsatellite repeats changed over time. Another nice example is the microsatellite typing scheme for Candida parapsilosis. So 10 years ago, we were asked to help a team from Austria. They had to deal with an apparent outbreak of Candida parapsilosis on the cardiothoracic ICU. And based on the Diversity Lab fingerprinting system, I provide here a small representation of that in the right upper corner. It's not the actual Candida parapsilosis Diversity Lab fingerprint. But with the Diversity Lab fingerprinting method, all the isolates of Candida parapsilosis from this specific ICU were identical, but they were a bit skeptic. So we were asked to set up and run Candida parapsilosis microsatellite typing. And based on that, you see that there is a huge diversity in the green bubbles. So each bubble represents a strain or two strains. And the larger bubble often includes multiple strains from the same patient. So at the end, there was no outbreak at all in this hospital. Recently established typing scheme was that for Pigia couchiliaceae, better known in the clinic as Candida krusei. So the clinicians prefer to use Candida krusei, and I think this is mainly due to the difficult to pronounce name of Pigia. My colleagues from Nijmegen, they applied microsatellite typing on a set of more than 100 Pigia strains. And with that, they established this approach, but they also run whole genome sequencing of a selection of the strains. And with that, they proved that microsatellite typing is as discriminatory as whole genome sequencing. So it's up to the laboratory which methodology to follow. It's either STR if it's more cost effective, or whole genome sequencing if that's fully implemented. Then the major topic, Candida auris, comparing typing techniques. So since 2012, 2013, several outbreaks of Candida auris appeared. And I show here the first two figures from the outbreak investigation. On the left side, an M13 fingerprinting gel, and on the right side, an ITS sequence-based phylogenetic tree. And this was the first outbreak published in 2013 from India. And since then, various techniques were used. mainly ITS sequencing, but also MALDI-TOF mass spectrometry, AFOP, multilocus sequence typing using 4 different genes, the well-known ITS, LSU, RPB1, RPB2 genes, and AFOP that is often done by our laboratory. But also microsatellite typing was initiated again by the team in Nijmegen. They also investigated hundreds of Candida auris strains from a global collection, and as you can see, there are some major clusters of isolates, and this is proven to be again a proof that it's a very rapid and cost-effective approach compared to whole genome sequencing or AFOP genotyping. So we compared the various typing techniques a few years ago, and well, ITS typing, it's not suitable. You can better call it ITS identification for Candida auris, because the 4 different clades cannot be reliably differentiated from each other. AFP typing has high cost, is rapid, but reproducibility is questionable. So you have to repeat the full analysis again. Every time you want to add extra strains, you have to do the whole data set again from scratch. So that gives the high cost. And microsatellite typing has low cost, is rapid and reproducible, as it generates a number, and the number you can put in a database, and from that point you can set up your own database to compare new Candida auris strains versus the old ones that were identified in your own hospital or even a global database. So the sequence-based typing methods are mainly multilocus sequence typing, and there are for Candida species 4 major multilocus sequence typing schemes. They are for Candida krusei, so the Pichia kutsirafsevi, Candida tropicalis, Candida glabrata, and Candida albicans. And the old MLST website has been taken over by NCBI, and I would like to forward you to that specific site, because it's a really nice site, but the disadvantage is that not everybody is deposits their data into this database. So my personal favorite nowadays is whole genome sequencing. There are 3 platforms commonly used. That is Illumina, or short fragment sequencing, with all the 8 different platforms. PacBio, long fragment sequencing with the different platforms, and the Oxford Nanopore technologies, with a whole range of machineries from very portable devices to the more larger machines. And for our laboratory, we fully focus on Oxford Nanopore technologies due to the relatively low costs and the ease of use. The disadvantage of genome sequencing is the huge amount of data that comes out of a sequencer. So the costs drop significantly over time per gigabase pair. And the disadvantage is how to deal with all the data. And during the past years, the manufacturers and especially Nanopore have provided tools to more easily run identification tools on their data. So here's a comparison between the different techniques for Candida auris. On the left side is a set of Candida auris strains that was analyzed by whole genome sequencing, and the figure next to it is the previously shown AFP fingerprint figure. And we were able in 2015 or 16 to rapidly genotype an outbreak from the United Kingdom in London in an ICU unit. And within 2 days, we had this AFP generated. And at that time, it was quite a challenge to sequence all those isolates. So that took a while before that was published. But the advantage then is, of course, that you have real sequence information, while with AFP fingerprinting, you have only your fragments and no sequence information at all. A few years back, a very large study was initiated, and they collected and they generated data for over 300 Candida auris strains from all over the world. And by using so-called SNP calling, they generated this phylogenetic tree that also showed that there are 4 different clades of Candida auris, and that's specific clades have only relatively few single nucleotide polymorphisms compared to the number of SNPs between the clades. A recent example of outbreak type of Candida auris is a case of a small outbreak in Salvador da Bahia in Brazil. It was in the middle of the Corona pandemic. And at that time, there were no flights to or from Salvador da Bahia. And they suddenly had an outbreak of a fully susceptible Candida auris lineage in their hospital. So we first applied microsatellite typing, but we were quite intrigued by this, because it belongs to the so-called clade 1, and this is from the mostly resistant lineage from India and Pakistan, so the southeast clade of Candida auris. And when we applied nanopore sequencing and run a SNP calling, we've observed that this isolate was fully basal to the clade 1. So these questions where the origin of Candida auris is. Because so far there are no other outbreaks or previous outbreaks or mentions of Candida auris cultures from Brazil. So it's really a question where and how this Candida auris appeared into South America. A nice one is Candida blanchi. So we see more and more cases of Candida blanchi. And like in Ratna's talk already mentioned, misidentifications are commonly seen, as certain rare species are not in databases, for example, in the AP20C system or in MALDI-TOF databases, or commercial chromokinic agars that are not tested with a rare species of Candida. So in this case, the Candida blanchi appeared to be slightly similar to Candida tropicalis, and it was misidentified as Stephanoscus siveri by the Vitek2 system, but it was also fluconazole resistant. And in India, they had to deal with a small outbreak in a neonatology ICU unit, and they used the SNP calling next to AFP fingerprinting to see how the relation was of all these isolates. And there was one isolate from this outbreak that appears to be not related, as there were close to 70,000 SNPs compared to all the other strains. So summarizing our current approach for fungal outbreak typing, first we know, of course, what the identity is of the species. So we first run AFP fingerprinting, and if that is not giving any answer on how, if it's very closely related, then we need to dive deeper into the sequences. So we first check if there is a genome sequence available, and if so, we check what the genome coverage is. It must be high, and also the sequencing technology. So we do not prefer short fragment sequences, but BacBio or nanopore data, as these techniques can sequence microsatellites, while this is quite difficult with Illumina sequencing. And we are of course interested in the microsatellites. And then we also look into the number of scaffolds. So that must be around 10 scaffolds for most of the Saccharomycotina yeast, so the Ascomycetes yeast. Based on that, we develop either a novel microsatellite typing approach, or we first go for de novo genome assembly followed by annotation. And subsequent steps are either in-house variant calling. So if you have only one strain, we can easily run in-house variant calling. And by many strains, we focus more on the microsatellite typing to have less costs. So in conclusion and take home messages, derivable somal DNA ITS marker is preferred for identification of yeast. And I would like to mention the ITS database from the isham.org website. Use different ITS primer combinations for the detection and identification if you use clinical material. So that is the ITS 1 and 4, and ITS 5, 4, and ITS 86 and ITS 4 prime combination. AFP and microsatellite typing are rapid tools to investigate multiple and large number of yeasts. Whole genome sequencing is recommended in the case of a novel yeast pathogen, for example, Candida filterna, that was mentioned by Ratna in the previous talk. Together we have sequenced the genome sequence of this, of the type species, and via that way we were also informed that at various other locations in the world, Candida filterna is popping up like Candida auris. Whole genome sequencing can be used to develop molecular typing by MLST or microsatellites. You can investigate resistance mechanisms, and what we use is variant calling or phylogeny. And on top of that, I would like to share also with you that please do not hesitate to deposit rare yeast in a culture collection. For example, our CBS culture collection, or that of HCC, CGUG, or any culture collection in Asia. It's for free, and with that, atypical strains remain available for researchers in the far future, and that can be really nice at that time when people deal with outbreaks of novel pathogens. And then lastly, the reference work, the yeast, is now online available. You can register and check the content of it via www.theyeast.org. And it includes all the species description, but also we are working on including antifungal resistance data and many more. And with that, I would like to acknowledge my team. And of course, I would like to thank you for listening.
Okay, thank you, Dr. Ferry, for the very interesting talk. Now we come to Q&A session. Is there any questions here for Juan Ratna and Dr. Ferry? There are some questions that we'll ask to Juan Ratna. I think some of them have already been answered. So for Dr. Ferry, this question is from Dr. Siti. She asked, which is the which ITS that you would suggest or prefer for use in clinical microbiology lab for PCR identification, and how do you choose?
So you mean which, I assume, yeah, ITS, and which primer combination?
Yes, correct.
Well, for strains, we commonly use ITS primers 1 and 4. So that spans the full ITS 1 and 2 regions. So ITS 1 and 2 are split by a very small gene fragments, the 5.8S, but we prefer to have both ITS 1 and 2. So therefore we use primers ITS 1 and 4. But from clinical material, we always use 3 different prime combinations. And most often only the small fragments with ITS primus, ITS4 and ITS86 are most successful.
Thank you. Any more questions from the floor, from the participants? All right, if there's no more questions, we can end this CME session. So thank you very much for your active participation. Thank you, Puan Ratna and Dr. Ferry, for your very interesting talk. You have enlightened us a lot in this field. And we will see you all next time. Thank you. And also thanks to Dr. Chiu for helping with the webinar. Thank you very much, Dr. Ferry. Thank you, Dr.
Thank you.
Puan Ratna. Thank you. Bye-bye.
Bye-bye.
Bye-bye.